Okay. I think everybody can see my screen.
Yes.
Yes.
Okay, great. Well, it's my great pleasure to have this opportunity to give a presentation at such an elite group of scientists, especially the women scientists. Unfortunately, we have to give this online. It would be great if we could be all together. It would be my third time in Cairo. Hopefully we can come again in another opportunity. Today I want to talk and before starting I want to thank to the organizing committee and Professor İlkay for this invitation. And I want to talk about the recent advances in vaccine technology in regard to sustained development goals. Currently, I am both working at the university and also at Turkish health institutions, and my main research area is vaccines. That's why I have chosen the vaccine technology advancements to share with you. And after giving brief information about vaccination and what are the unmet needs for vaccines, I will give very short examples of new adjuvant technologies, and then before concluding my talk, I will give a summary of the work my group has been performing at Hacettepe University. As we all know, vaccination or immunization is performed to stimulate the body's immune response against the pathogens to protect the person against that infection or disease. And for this purpose, we administer the antigens. I will tell you how we prepare the antigens. And then when we inject them into the body, our immune system is activated and antibodies are produced, and they remain in the body. And when we are exposed to this pathogen again, as we have the antibodies against these pathogens, our body is protected from the infections. Here you can see my grandson. Also, he had posed for this slide. And as SDG goal, it is also important to support the research and development of vaccines, and especially for the developing countries or LMIC countries, and especially to survive the lives of millions of children. And we know that vaccines, after sanitation and clean drinking water, is very important for the lives of people. And with vaccination, we can decrease the health burden which is caused by the infectious disease. And of course, this has impact on the economics. And also, because by vaccination, we can prevent the diseases, this will result in a decrease in antibiotic use, and subsequently, we can decrease the antimicrobial resistance incidence, which is becoming very important in the world. And recently, in October, the One Health Trust, in collaboration with the International Federation of Pharmaceutical Manufacturers and Associations, they have released a report, and they have shown the evidences that the vaccination does slow the antimicrobial resistance. So we have many vaccines, but because of their efficacy and because we need long protection, some of these vaccines are needed to be improved, or some of the vaccines are not available at the moment. For example, we have the malaria vaccine, which has only 30% efficacy, but as there is no other vaccine, this vaccine was improved in 2015 with a special approval procedure. I just want to give you a short story of how a vaccine is developed and how long it takes to develop a vaccine. It's not easy. Here, as an example for malaria, in 1880, the relation between the malaria and the parasite which is causing was discovered. And almost 100 years later, the vaccine studies had started. And as you can see, after 1980, several studies have been conducted in vivo, in animals, and also in clinical studies. And eventually, in 2015, this vaccine called RTS was approved to be used only in United Nations listed countries with a special procedure called EMA Article 58. And later, WHO recommended the use of this vaccine in many African countries. And eventually, in 2022, this vaccine was contained and accepted for the pre-qualification. And recently, in this year, in October, a new vaccine has been recommended by WHO for malaria. As you see, we have this malaria disease for a very long time, but the vaccine history is not very long, and still there is not a real marketing authorization for a malaria vaccine. In the case of COVID-19, this was a bit different because it was pandemics and we needed the vaccine urgently. And I just wanted to give this period of how we identified the SARS-CoV-2 vaccine virus, and then it was in late December 2019, and then the next year when the pandemics was announced, the vaccine studies initiated, and in December 2020, so almost in 12 months, the first vaccine received emergency use approval. And in 2022, last year, these vaccines started to get usual conventional approval. And when we look at this year, these vaccines with new strains have been approved. But this is not the usual way. So this is very, very exceptional. Do not think that every vaccine from now on can be developed and get into the market in such a short time. That's not possible. And WHO has defined a target product profile for the vaccine. We need to know for which disease this vaccine is needed, and we need to know the target population. Also, when we are vaccinated, as you know, sometimes we get several doses. For vaccines, we call it boosters. So we need to define the exact dosing regimen for the vaccines. And we should know how long this vaccine can protect us. For example, for COVID vaccines, at the beginning, nobody knew about the protection. And then later we saw that for some vaccines, it was only for 3 months, and for some vaccines for 6 months, but that's not what we really want. Most of the vaccines are protecting forever, or some protect for 10 years. So this is very important when we are designing a vaccine. And route of administration is important because when we want to have mass vaccinations, and if it is a parenteral route, then you need the syringes, and that means contamination, because we don't want to contaminate people while we want to prevent these people from the infections. So generally, the vaccines are administered through parenteral routes, but many studies are now focusing on for alternative routes, which we call non-invasive, like oral, but because of the stability of the vaccine antigens, that is almost not possible for the moment. We have very few vaccines which are available for mucosal delivery. And of course, the most important issue for a vaccine product is its safety and efficacy. We need to meet the global regulatory requirements for safety and efficacy. Another criteria are product presentation, storage, and shelf life. For example, before pandemics, nobody could imagine giving approval for a vaccine which has to be stored below 70 degrees, but now because of the pandemics it was possible. But for the new vaccines, again, this should not be the case. And also, when you are manufacturing a vaccine, the cost of the goods, the manufacturing scale, and the type is very important. So when we prepare the vaccines, this is the pathogen which causes the infection. We can use it live but attenuated, or we can kill this vaccine inactivated. But when we use the inactivated vaccine, it may have side effects. Therefore, we use the subunits of this vaccine. As you can see, we take some subunits which are effective, which can be recognized by the system, or we can highly purify these antigens, or we can use the genetic codes. But when we purify these vaccines, the immunogenicity can be decreased, although we have higher safety. So we need the adjuvants. So in recent years, the adjuvant technology became very important. Adjuvant in Latin, it means to aid. It comes from the word adjuvare, and it is used to enhance or modify this S is extra immune response. And for adjuvant and delivery systems, when we look at the adjuvants, we can classify them into two classes as delivery systems and immunostimulators. And also we can combine these systems. And recently, when we look at the new adjuvants, they are not only one molecule, but they are the adjuvant systems. And as you may notice, they are lipidic based delivery systems, and or virus-like particles which mimic the virus or which mimics the bacteria. And there are other formulations. As you may notice, they are all in particulate forms. Why particulate forms? Because here in this scheme you can see the vaccine delivery systems, and in this scale you see the size in nanometers, and here you see the pathogenic agents. So when you compare the size of the viruses or bacteria, and here you can compare the size of the delivery system. So we want to mimic the virus or the bacteria, so we want them to be taken up by the immune system. Among these delivery systems, we use liposomes. Liposomes, their structure is similar to the cell membrane. They have a phospholipid bilayer, and with a hydrophilic head and a hydrophobic tail, and they contain phospholipids and cholesterol, and especially the cationic liposomes are preferred because they are taken up by the cells easily with the cationic surface charge with positive surface charge. We can have unilamellar or multilamellar, and currently the vaccines I have mentioned, the first vaccine for malaria, it is based on liposomal structure with also additional adjuvant immunostimulant molecules. As we all heard about lipid nanoparticles with the mRNA vaccines, they are similar to liposome structure, but here you can see with the liposomes inside we have more aqueous medium, but with the lipid nanoparticles, this is more non-aqueous, so we can incorporate the mRNA, the RNA unit or genetic code into these lipid nanoparticles, because otherwise the mRNA degrades at room temperature. So for stability, both in vitro and in vivo in the body, we need this till it is taken up by the cells. And we can also use specifically targeted ligands like pegylation, polyethylene glycol to target. So these lipids used are generally cationic lipids. And currently, the Moderna and biotech Pfizer COVID vaccines are based on lipid nanoparticles. And another particular structure, again lipid-based, is ISCOMs. When we call this is immune stimulating complexes, ISCOMs can be with antigens, and if we don't incorporate the antigen during preparation, we call it ISCOM matrix. And these ISCOM matrices can be physically mixed with the antigen during administration. And the recent adjuvant developed is matrix M, and currently this vaccine, another COVID-19 vaccine, contains this matrix M adjuvant. It contains different fractions, fraction A and C, of saponin in a liposomal structure. Another carrier system and also adjuvant is virus-like particles. Here, particulate antigen, it can either we can prepare this with the antigen itself, or we can prepare from a different virus and incorporate with another antigen. And currently, FDA and EMA approved human papilloma virus, which is based on virus-like particles, but they are not enough to increase the immune response. Therefore, they need additional immunostimulant adjuvants. So combination is needed. In the coming tables, I try to list the adjuvants approved with the product. Although there are many studies conducted on adjuvant development, currently there are only 7 adjuvants available on the market, and they are, when you look at their composition, they are more or less similar. Because of the safety issues, it is not easy to have an adjuvant reach to the market so easily. Here you can see MF59. I'm not going to read all the content due to the time strict. These are adjuvant system 03. As you can see, it has also squalene, but it has additional alpha tocopherol in it. But this was for the pandemics during the influenza in 2009, but it is not available in the market anymore. So the company did not continue with this adjuvant. Another adjuvant, and most of these adjuvants, as you can see, Novartis was sold to GSK. So these adjuvants belong to one company. So the market is also very limited, the vaccine and adjuvant market. And here, these are MPL, monophospholipid. This is also a bacterial-based adjuvant with immunostimulant activity, and also they are combined with aluminum salts. And another
could you wrap up in about 2 minutes, please?
Yes, I will do that. These are the unilamellar liposomal vehicles, and they were also approved, but they are not on the market anymore. And with this malaria, again, an adjuvant, as I mentioned, liposomal structure. And recently, the immunostimulant-based adjuvants have been developed. As you can see, these are the immunostimulant adjuvants, and I have mentioned about matrixav. Very shortly, in 2 minutes, I will just mention about the studies what we have done. We have used chitosan, which is obtained from crustaceans' shells. First, you get chitin, and then you get the chitosan. I think most of you are familiar with this compound or polymer. And we use this as a delivery system, also for its immunopotentiator activity. And because it has penetration enhancers, it has an additional activity as a vaccine delivery, especially for mucosal and dermal delivery of vaccines. And first, we started with ovalbumin. And we look whether our chitosan microparticles are effective. We, in collaboration with CSIRO in Australia, we combined chitosan with ovalbumin in gel and sponge form. And in sheep, we have administered nasal and rectal, and we have shown that it induces the immune responses in presence of chitosan. Similarly, with tetanus toxoid, we used chitosan and its derivatives, and we have administered nasally in mice and obtained very promising results. And also another veterinary vaccine, we used alkaloid. That was conjugated to a protein. And we have, this is used for the jaundice, for the animals, because when they eat this plant, they get jaundice. So that was against this alkaloid. And we again tried it nasal and rectal in sheep. And then we have used also.
Yeah, I'm afraid time is up. If you could just conclude, please.
I will. So it is foot and mouth. We have waited for half an hour to enter this session. So I guess I can have 5 more minutes if you allow me. So foot and mouth disease.
No, I'm sorry. No.
I stop.
Yeah. But maybe you could just, okay. Well, thank you very much. Yeah. Thank you very much.
Congratulations. You have done a very good job. But anyways, we are very sorry because the time is allotted only for 20 minutes at least. Even that also you have exceeded a little bit. But vaccination is very, very important.
I waited for half an hour to start this presentation, but you didn't even give me 5 minutes, extra 5 minutes. So this is not what we are used to. I am in this area for 43 years.
Yeah, I know. It is with your vast experience. I know it's very difficult also to conclude such a big assignment, especially the lot of work is being carried out on the vaccination. When you want to achieve the sustainable development goals, pharmaceutical science is very, very important because mainly to protect the health, to take care about the health that we all know very well. It is very difficult to give the very essence of your experimental results in a short duration. We are anyway sorry, but you have done a very good job. Congratulations. Maybe if time is there, we will really discuss about it. Thank you very much.